So we're done talking about, oceanography stuff for the most part, and plants, okay? And now we finally get to talk about animals, okay? But there's a lot of different types of animals out there, and not all of them are interesting, so we're gonna talk about the boring ones first. Okay, that's why I have these two completely unrelated organisms in the lecture, and it's also not logical, right? So, worms is actually a pretty advanced organism compared to some other ones, so, it's not in a evolutionary progression, but, we just want to get the least interesting ones out of the way and talk about the more interesting ones later, right? Like crabs. Once we get into that one, I bet it's going to be more fun. But today we're going to talk about these, all right? Porifera and worms. And what are these things? We know what a worm is, or we think we know what a worm is. Maybe we don't know the formal definition, but I'm sure you guys all have seen a worm, right? It's shaped like a small snake, right? A little thing, wiggly thing on the ground. What is this? What is that? You know what that is? Have you ever heard of that word before? It's okay. We're going to find out, right? This is what it is, right? What is this? Sponge. It's a sponge, but there's a difference between this sponge and your sponge. This is a real sponge, right? This is an animal, okay? This thing used to be alive. Okay, back in the olden days, this is what people would use to do sponge things, like, soak in water, wash dishes, right? Nowadays, sponges are all synthetic, but, you can still find these things at the beach, and they're pretty cool because you pick it up, and it's like squishy, and I'm like, whoa, this is actually an animal. It wasn't man-made, they're actually squishy like that. Okay, so sponge, right? Sponge is a type of animal, and sponges are in the phylum Porifera. Why? Because you guys see all these holes? All right, those are pores, right? Right, all the pores that you guys see, the holes, that's why it's called Porifera. Pores, pores, little holes. Okay, the scientific term for those pores is, well, it's hard to see, but ostia, okay? O-S-T-I-A, ostia. Okay, that's the scientific word for the pores on a sponge, okay? So let's learn that word. Okay, so porifera, right? These guys all have these holes. What are they for? Well, we'll find out, right? But I'll give you a hint. Water goes through those holes, okay? Why does it do that? I don't know. Important life processes, maybe, right? Okay, so if we look in the microscopic scale of a sponge, we'll find cells that look like this, right? This is a sponge cell. It's called a choanocyte, which means color cell, okay? Choanocytes. And they all have this tail, right? This tail is called a flagellum. Have you guys heard of flagellum before? Yeah? Do we have a cell that has a flagellum on it? Yeah, sperm cell, exactly, right? A sperm cell has a flagellum on it. What does the sperm cell use its flagellum for? To swim around, swim up the fallopian tube, right? That's what the sperm cell uses its flagellum to do. Okay, so think about this for a second. What if you didn't allow the sperm to move? Then what do you think the tail would do? What do you guys think? It would, exactly, move the liquid around it, right? That might be a little hard to visualize. Let me translate that into something else, right? The tail of a sperm, right, that helps it propel it forward is the propeller of an airplane, okay? So if you turn on the propeller, the airplane goes forward, right? What happens if the airplane has its brakes on, or otherwise you don't let the airplane move? Then the propeller is just a fan at this point. You guys see that? What do fans do? They just move the air around. You guys see that? So like, let's just say that you had this choanocyte and it had this tail and it can swim around, but if you like stopped it from moving and it keeps beating its tail, then it'll end up moving the water around it. You guys see that? Right? It'll end up moving the water, and the combined forces of all the choanocytes moving the water will create a little current. Right? That's what it's trying to do. Okay? So the whole point is to beat the water around it, all right, for the purpose of getting it through its body, so that it can do a process known as filter feeding. We barely mentioned filter feeding yesterday, but we were talking about mussels and how they do filter feeding to do what? To eat. What were they trying to eat? The phytoplankton, right? Like the diatoms and stuff. I mean, that's how the poison got inside the muscles, right? So sponges do filter feeding, right? That's their feeding style. Sponges filter the water to try to get the plankton inside or the other particles inside. And they do that by passing the water through their body, and then they catch all the stuff inside. Right? So let's see what the choanocytes look like in context of the whole sponge, right? So here's a sponge. You blow up the wall of the sponge, and it looks like this right here. See all these little choanocytes? All the choanocytes beating their tails in unison right here, creating a little current. That current causes the water to get sucked in through this. What is that? Oh look, it's the ostia, right? You just learned what that meant. What was the ostia again?
Pore.
It was the pores. Judging by this diagram, how big is the ostia? It's really, really small because look, it's in this picture at least. I don't know if it's drawn to scale, but let's just pretend it is. This ostia is as big as a color cell. One cell, that's microscopic, right? So like these, at least in this picture, this pore is tiny, right? So it turns out the pores are tiny, and the water gets sucked in here into the middle, gets filtered, and expelled through the big hole on the top. The big hole on the top is called an osculum. All right, so there's a difference. Right? The big holes that you guys see at the top of the sponge is called an osculum, and the little pores that you probably can't see on the sides are called the ostia. Those are the pores on the sides, right? Okay, so that's what a sponge does if it wants to eat. If a sponge wants to eat, get the little plankton out of the water, it's going to beat its choanocytes, get the water to go inside the pores and out through the osculum. Right? You can actually follow or trace the movement of the water if you dyed the water, right? You could squirt a little dye. Like, let's watch this video. This is a cool video, right? I mean, when I was a kid, I thought it was cool. Now I'm kind of bored of it. But like, you get these sponges and these divers, right? What do? They take this
toxic dye called fluorescein. By squirting it around the base of some sponges, we can observe how the water is moving by watching what the dye does. Within only seconds, the dye is pumped through the sponges along with the water.
All right.
As you can see, a sponge is a pretty good water
in
and also
from the sides,
strainer.
and it came out the top. What are these called again?
Won't come back out through the osculum.
Osculum.
Tube sponges are even more
Okay, we all got it, right? Okay, so the ostia, which, could you see the ostia? No, you couldn't, right? The microscope, it just looked like a sheet, but they squirted around the edge, and somehow the water went inside, and it came out the top, right? It was doing filter feeding, okay? So the sponge filter feeds by using its choanocytes to bring the water in. Okay, so water pumps in, comes out the top. Okay, so let's take a look at this. This is the biggest sponge in the world, right? You find this on the Great Barrier Reef. People like to take pictures in them, but it's not recommended because, I mean, sponges, they're like spongy, right? You can break them and stuff. Okay, so this barrel sponge has a big hole here. Where are its pores located? Is this the pore? No. So where are the pores? They're around the outside. You can't see them, right? The pores are over there, and the big hole that you see on top, what is that? That's called the osculum, right? And so the water comes in here, and it goes out the osculum. Okay. All right. So, therefore, all of these big holes that you guys saw, it's kind of misleading, right? You guys look at it as like, okay, a sponge has holes, it's porous, right? Because porifera. But it turns out all these holes are not the pores that we're talking about. All these pores are the oscula, right? Where the water comes out of. And the actual pores are microscopic. Okay, so let's look at this picture that shows you where the pores are. The big holes on top, all these holes that you guys see are actually the exit holes, right? The oscula. Whereas the tiny little pores that you guys see all around, those tiny little dots. Here's the blown up image. 0.5 millimeters. I guess that's pretty small, right? Okay, so the actual pores, microscopic. really see them, right? If you see a sponge, right, and you see all the holes in it, you're like, oh look, it has a lot of holes in it. It's got to be a sponge. Those are its pores. Not quite. I mean, it is a sponge, but those holes that you can see are the oscula, right? That's where the water comes out of. Okay, so this is a little distinction that we can make. Okay, so now that we're talking about skeleton, we can talk about what the skeleton is made of, right? Take a look at this sponge. This is a sponge, or at least scientists classify it as a sponge. Okay, that doesn't really look really spongy. It doesn't look very comfortable to feel. What do you think that's made out of?
Calcium carbonate.
Yeah, good guess. Almost everything's made of calcium carbonate. But this one.
Maybe your actual an average skeleton, like your bones, or
No, there's a skeleton of this sponge.
oh.
Yeah, not of humans. Okay? This sponge, its skeleton is made out of the other one, silicon dioxide. It's silicious. You guys see, it kind of looks like glass, maybe, just a little bit, right? So this is a glass sponge, right? It's made out of silica, right? It has silicious spicules. And here we can introduce our first member of the taxonomic level of class, right? We've been talking about phyla so far, phylum Porifera, but now we're going to start talking about classes, okay? So under phylum in taxonomic level we have classes, and we're going to go over 3 classes of Porifera. The first one is quite the mouthful, all right? Hexactinellida, right? You guys try to say that, Hexactinellida. Okay, well, the first part was a good shot, but maybe you guys can work on the last part. If you guys practice how to say these things, then the spelling would be a little bit easier, right? So hexactinellida, these sponges are made of silica, right? So if you touch it, is it going to be spongy? No, it's going to be hard, right? Because glass and quartz and stuff, that stuff is hard, right? So class hexactinellida, a hard sponge. Now let's look at this sponge right over here. Class Calcarea, a second class. This one, do you think this one is spongy? Well, it looks a little bit more spongy than the other one. But judging by its name, what do you think it's made out of? Calcium carbonate. Okay. Is it hard or spongy? This one is hard. Calcium carbonate, right? Seashells, right? Another hard sponge. Right, this is interesting. Not all sponges are squishy, right? 2 out of the 3 of the classes that we're going to talk about are hard sponges actually, and these ones are made of calcium carbonate, right? They have calcareous spicules. Okay, so when are we going to get to talk about the actual spongy ones, right? Well, Calcarea and Hexactinellida are not the majority, right? They make up roughly 15% of all sponge species, the rest of which are in another class, right? Like this. These sponges, the one that are actually squishy. Those ones. Demospongia, right? Demospongia over here. That is the 3rd class that we're going to talk about. And these ones, they do not use spicules, therefore they are not hard to the touch. They're soft and flexible, right? So the barrel sponge, soft and flexible. If you press down on it, it would, you know, it would sink. I don't know how to describe it. It would like cave in, right? Okay. Is there anything on this page that's not a sponge? Yeah, a little jellyfish, right? The jellyfish is not a porifera. That one we're not going to talk about until next week. Okay, so yeah, they have an elastic protein called spongin. That's what makes them spongy. And I'm not completely sure what the purpose of being spongy is, but it doesn't really matter. The important part is that it has coanocytes that can suck in water. It's going to do filter feeding on it, and then it's going to expel it through the oscula at the very top. Okay. All right. Any questions so far? We're all good. Okay. So now let's introduce the concept of larval type. Okay. So when organisms grow up, we can study its life history, which means what does it do from birth to its death, right? That's its life history. And when most things are born, they look like something else that's completely different from its adult, right? When we're born, we look more or less the same, okay? So our larval type is actually inside the womb, right? It's called an embryo. But there's other things whose larval type, when they're born, right, they look substantially different, right? I'll give you an example right here. The caterpillar, right? The caterpillar is a larva of a butterfly, okay? So it looks substantially different, and then it goes through a process known as metamorphosis and becomes the adult form, okay? So most of these invertebrates that we're going to talk about throughout this class have a larval form that is different than the adult. But here's the thing, not every phylum has a similar looking larva. Actually, I shouldn't even say that. I should say all of the different phyla have a different larva. Okay, so we call it a larva type. And the larva type of a sponge, right, if we want to figure that one out, we're going to have to look at the life cycle. Let's take a look at the life cycle. Here's a life cycle. Let's just say you have a young sponge. The young sponge grows up into an adult sponge, right? When it grows up into an adult sponge, it kind of acts like a hermaphrodite, and it sends sperm to another sponge. This sponge sends sperm back, and they co-fertilize each other, all right? And then when you have the baby and the egg and all that, it comes out and hatches into the larva. This is the larva, right? See, this looks substantially different from this. Okay, so this is the larva type, and it is called a parenchyma, another hard word, okay? So I can challenge you guys right now. Let's all say parenchyma together. Parenchyma.
Parenchyma.
Yes, parenchyma, all right? It's another hard word to spell and to say, but if you guys practice at home, it shouldn't be too bad, right? So parenchyma larva, what is it? It's a larva of a sponge that looks like that. Basically a ball with a bunch of flagella around it. Okay, where did it get these flagella from? Well, look at each of these cells. What are each of these cells? They're the choanocytes. All right, each of these cells is a choanocyte. Okay, so you get just a ball of choanocytes, and that's basically the larval form. It's super simple, right? So, you know, looking at the adult sponge is just a bunch of choanocytes collected in the shape of like a vase. And then the larva sponge is just a bunch of choanocytes collected in a ball, right? A sphere. Okay, so we're kind of getting the idea that sponges are an extremely simple organism that's just made out of a bunch of choanocytes, right? Well, that's really interesting because of that fact. We're able to trace back the history of the sponge and determine that it was one of the first animals, if not the first, right? Let's look at this. The main sponge cell is the choanocyte, which looks a lot like, strikingly similar, to a choanoflagellate, which is a type of protist, right? We remember what a protist is, right? So, based off the definition of a protist, what can you say the body of a choanoflagellate is? That's it, right? That's the whole body is just this. So a sponge is made out of a bunch of those, and a choanoflagellate is just this by itself. So maybe if you can envision sometime in the history of evolution, a bunch of choanoflagellates got together, started working together. It's basically a sponge at that point, right? A collection of those things. Okay, so it's super simple. Like, we're not just a collection of cells. We are a collection of cells that each part of our body has a different function, right? We have specialized cells for this and this and this, but for a sponge, all of its cells are these, right? It just took a bunch of these and put them together. That's it. It's super simple, right? So, tracing back evolutionary origins, right? Choanoflagellates are here. This is not an animal yet. This is like the link between animal and non-animal, right? And then here are all the animals. Sponges are probably the first animals that ever came out, right? I would argue that it's the first animal that came out. Some scientists with some, molecular data have suggested that we have ctenophores, the comb jellies as the first animal, but I don't know. A comb jelly has a really interesting property, right? It has some sort of symmetry to it, whereas the sponge does not. All right, we'll get into symmetry in a second. But the point of this is just to show you guys that sponges are super simple. They're just a bunch of choanocytes. A choanocyte looks like the protist choanoflagellate, and it's probably the first animal, right? The difference between animal and protist is animals have multicellular and protists single cell, right? That's literally the difference. Okay, so sponges and comb jellies are amongst the first animals to ever. Okay, but yeah, like I said, the comb jelly looks like it has some sort of symmetry to it. The sponge has no symmetry, right? It is asymmetric, right? Asymmetric. Let me write that word down because there's an important root that I want you guys to see, or a prefix. Asymmetric, right? A means without, right? Or no. Okay, so we're gonna see some other words that start with A, and you'll realize it means the opposite, right? It means no or without. Okay, so sponges are asymmetric. Symmetry has not evolved in the animal kingdom yet. Okay, so when we move a step forward in evolution, you'll see that symmetry evolves really quickly because symmetry is the fundamental basis of organization, and the more organized you are, the better, right? So, organization, right? Symmetry, like this. Do you see there's symmetry in here? There's no symmetry in here. Right? That's just a amorphous or irregular shape. But this is symmetrical, right? Symmetry means there's some sort of pattern to it. You can find mirror images throughout, right? This type of symmetry, like a circle, like a car's tire, is called radial symmetry, right? Radial for radius, right? You guys know what a radius is, right? What's a radius?
Half of the diameter.
Yeah, it's the half of the diameter, right? So it's like from the center of the circle to the outside, right? That's what a radius is. And, you know, these things have radial symmetry. I like to describe radial symmetry as branching outward. Okay, so if you ever want to describe radial symmetry, someone needs to say it's a shape that branches outward in all directions. Okay, radial symmetry. In contrast, here's another type of symmetry. Right, look at this. Worms, right? This is our segue into worms now. Worms all have a different type of symmetry. Do you see it's not radial? It doesn't branch out in all directions. I put humans on here, not because we're a worm, but we have the same type of symmetry as a worm does. Bilateral. Right? Bilateral, which means two sides, right? Two sides, because you can cut it in half, and you'll get a mirror image, both sides, right? You can cut a person in half. You get a mirror image on both sides. All right, so we just learned a couple different symmetry types. We got radial symmetry, and we have bilateral symmetry. Right? Different organisms have different symmetries. Later on, we'll find out which one is better. Yeah. Bilateral symmetry. All right, so let's do a comparison, right? Comparison: radial symmetry versus bilateral symmetry. Well, looks like radial symmetry, you can cut it in any which way, and you'll always get two halves. Right? I put 4 lines of symmetry on here, but I could draw a couple more, couldn't I? How many lines of symmetry do you think it has? How many? How many of these lines can I draw and cut in half? Infinite, yeah. Right? Radially symmetric things have infinite lines of symmetry. How many lines of symmetry does a bilaterally symmetric organism have?
One.
Just one, right? Just one line of symmetry down the middle, cutting into 2 mirror images. That's it, right? Why is this significant right here? Right? Bilateral symmetry. You can only cut it one way, and you get 2 halves, right? Well, one of the benefits is you can have a through gut, right? What's a through gut? It's basically, you know, a digestive tract. And a digestive tract is, I'll just draw it real quick, super simple. It's a mouth and an anus and a tube that connects it. That's the digestive tract. All right? Put some flesh around it, and that's a worm, right? The worm is the easiest shape to have a digestive tract, right? A through gut, right? It's hard to have a through gut when you're radially symmetric. When you're radially symmetric, you'll find out that it has a one-sided gut. There's only one hole, right? We'll find out what happens when you eat. You only have one hole. You eat up, right? But you know, like us, the worm shape is the easiest form to have this digestive tract. That's why worms were the first organisms that evolved bilateral symmetry, right? Bilateral symmetry evolved, and you know, it could have been any of the shapes of any of the bilaterally symmetric organisms we saw. However, the worm shape is the easiest. Remember, in the grand scheme of evolution, the rule of thumb is the easiest things come first, right? The simplest things come first. That being the worm shape. The worm shape is the simplest thing that came first. Just a little bit of meat wrapped around a digestive tract. Okay. What's another perk of bilateral symmetry? If you have bilateral symmetry, you have a front and a back, right? In anatomical directions, it's called anterior and posterior, right? Anatomical positions. Anterior means front, posterior means back. You have a front and a back. Why is it important to have a front and a back? What do you have? What body part is in the front of your body? Your mouth, yeah. But I mean, what's your mouth on? It's on your head, right? Having a head is probably the biggest perk of being bilaterally symmetric, because if you have a front and a back, then you can have a head. You have a head region and a tail region, right? The head region is the anterior, and your head is super important, right? We take it for granted because we have a head. All the animals that we interact with have a head. We don't really think of animals that don't have a head and how much they lack, right? We think that this is how it normally should be. But we have so many advantages because we have the head, right? So let's talk about those. If you have a head, then you have a brain, right? You have a brain inside the head, a central nervous system, right? And if we have a brain there, then we have a lot of things associated with the brain there, such as your senses, right? All of our senses are concentrated in the head, right? So we have a head. The head has a brain in it. And all of our senses are there, right? Let's just check them off. Our eyes, vision, we got it right there. Hearing, right here on our head, yet again, right? Our nose for smell is on our head. Our tongue for taste is on our head. And, well, for touch, our fingers are number one, but number two are our lips. So we still have everything on our head, right? All of your best senses are on your head. Why are they on your head? Well, for a couple reasons. One is, if they're on your head, then they encounter the environment before the rest of your body encounters the environment. Right? That's really important, because what if part of your body encounters the environment before your senses encounter the environment? So before you can see what you're gonna touch, you touch it. What if it's something dangerous, right? But thankfully your senses encounter the object before the rest of your body do, so you know whether to touch it or not. Right, you know whether to go there. Okay? All your senses concentrated in the front so that your senses encounter the environment before any other part of your body does. Right, what's another reason? Well, all your senses are close to the brain. Right? If they're close to the brain, then it's much more efficient because the brain signals don't have to travel as far. Right? Imagine if you had eyes on your hands or something, you could still see out of them, but you would have a slower reaction time because you gotta send the signal to your hand, right? The reaction time is decreased, but thankfully, all of our senses are right next to our brain and reduces the time. Okay. Keep in mind, all these things is due to the fact that bilateral symmetry. Bilateral symmetry affords front or back, a head, a brain, senses concentrated in the front. Right? Okay. Here comes a difference between radial and bilateral symmetry. Radially symmetric organisms, they don't really care what direction they're facing. They're the same direction all around. So they're kind of mediocre at all directions, whereas the bilaterally symmetric organism is concentrated solely in front. Okay, so if you concentrate solely in front, then you sacrifice all the other directions, right? Just look at yourself. You can't see behind your head. You can't see to the sides very well. You can only see in one direction, forward, right? Why? Because you've chosen, evolutionarily, to sacrifice all your other directions for forward, all right? And just be hyper efficient in forward. You realize that you don't need the other directions because if you ever want to see behind you, you just turn around and that becomes your new forward, right? So bilateral symmetry, having a front, is better than treating all directions the same. If you treat all directions the same, then you're going to be mediocre in all directions because you have to spread it out, right? But this way you can concentrate in one direction and be super efficient in that one direction. And if you ever need another direction, just turn to that direction. And you got it. This is true for senses, and it is true for movement, right? Your movement only works in one direction, right? If you wanted to get away from a predator, would you choose consciously to run backwards or sideways? No, you would always run forward, right? If you needed to run behind you, you turn around and run forward, okay? Why don't we run in the other directions? Because we're not very good at it, right? We sacrificed our movement in all directions except for forward, right? All of our other movement directions are not very efficient, but we are very good at going forward. Right? That's what bilateral symmetry does to us, right? We concentrate in one direction, we become very efficient, but then we sacrifice all the other directions. We don't need them anyways, right? Okay. Maybe you guys appreciate the fact that you're bilaterally symmetric in this one, right? And we're not radially symmetric. Okay, any questions so far? We have those. Okay, so yeah, now let's get into talk about some bilaterally symmetric organisms. I know I mentioned radial symmetry, but I only mentioned it just because we're on the topic of symmetry. We will revisit radially symmetric organisms on Tuesday next week. Right? For now, we will go on to talk about some worms. Worms being the first bilaterally symmetric organisms, right? Worms. These are worms. A lot of different types of worms. All these worms are in a different phylum. Isn't that intimidating? Maybe not. Maybe you guys recognize one of these worms, right? That's an earthworm, right? That's a terrestrial example. The other ones live in the ocean. Okay? But all of these are worms. Right? We call them worms not because they're related. No, these are not related to each other. They're in different phyla, remember? Just like chlorophyta and rhodophyta are in different phyla, right? These things are in different phyla as well, so they're not super related. They just look the same. They just look like a worm, so we just call it worm, right? There's a lot of there's a lot of words that we use to describe things just because they look like them, but they aren't actually them. All right? So worm is one of them, right? We call these things worms, but they're not all related. Okay, so let's go through each of these worms and talk about them just a little bit. We're going to concentrate on these worms. These are the most important ones. But the rest of them, I would like you guys to learn the name of the phylum and attributes about them that allow you to tell them apart from the other ones. That's it. Okay? Nothing crazy. All right? We're going to learn the name of the worm, and then we're going to learn how to tell it apart. Starting with the one on the left. The one on the left, that's a flatworm, right? Platyhelminthes. Platyhelminthes is the phylum known as flatworms, right? So these worms are flat, and you can tell them apart from other ones. Well, first of all, they're flat. But second of all, in their head region, they got what looks like eyes, right? They're not true eyes like your eyes, right? Your eyes have, you know, a lens, right? They have like the vitreous fluid, and they have a retina in the back that collects the light. Okay, so we have all sorts of things. This just has, it's just like a retina, right? Just some light-sensitive cells in the shape of a cup, right? So we call those eye spots, right? Eye spots are light-sensitive cells that collect light. They're not amazing for vision, but they're better than not having it, because with light-sensitive cells, you can at least tell the difference between light and dark, which is a lot more useful than you think, because maybe you can't see anything, but you can tell when it becomes light and when it becomes dark. If it becomes dark all of a sudden, maybe you've just crawled into a cave or something, right? Maybe there's a predator hovering above you. If you had no sense of that at all, then you just get eaten, right? So, you know, having the ability to tell light from dark is actually not that bad, right? It's actually pretty impressive, at least for this worm right here. Okay, so yeah, they got eye spots. They're flat. Platyhelminthes is a flatworm, right? Okay. You guys heard of a tapeworm before? Where does it live? I know where it lives. Like inside there. Well, mostly dogs, right? But I mean, they live inside mammalian intestines, right? Digestive tracts and then like parasites. Okay, that is a platyhelminthes, right? A flatworm. Okay, has anybody seen this show before? It's old, right? It's super old. You guys might have not even been born when I watched this. This was like before 2000. Yeah, okay, so I watched this when I was a kid. And I saw this one episode about the fluke man, and oh my god, I was so scared, okay? I believed in this stuff. Like, I thought this was real. Okay, this guy, he got this fluke inside of him, and he turned into this thing. I actually thought this was real. I was so scared, and I tried to avoid water and stuff. And these organisms, I looked up fluke in the dictionary because I was, you know, I was afraid of that stuff. But, you know, now that I know, it's just based off of platyhelminthes, right? I mean, they don't actually do this, right? They might be parasites, but they're not going to transform you or, like, you know, rip your skin up or something like that. That's crazy stuff. Okay. So, yeah, platyhelminthes, right? Okay. What is actually cool about platyhelminthes, though, is this is actually cool and like real stuff, right? If you cut them up, they will regrow, okay? Not just like if you cut off its tail, it'll regrow a tail. No, not just that. The tail that you cut off will regrow a head. And now you have more than one platyhelminthes, okay? The fact that it can regrow back a tail when you cut it off, that's called regeneration, okay? And some organisms can do regeneration. That's a really strong, you know, form of regeneration. We cannot do that strong form of regeneration. If you cut off a limb, then we lose a limb, but we are capable of regenerating skin cells, right? We can grow scar tissue over a wound, right? These guys can regenerate entire organs and entire like brain, like their head, right? And so in doing so, now that you have multiple, then it's not just regeneration anymore. It's reproduction now, right? Because reproduction means you end up with more than you started with. So, did it have to breed to do this? No, it's just by itself, right? No mating involved. This is a form of asexual reproduction, right? Asexual. What does the A mean? Without, right? Without sexual reproduction, they just reproduce by themselves, right? It's pretty convenient. They make a clone, right? 2 clones. Okay, so a lot of different organisms can do regeneration. Not all organisms have regeneration that is this strong, right? Strong reproduction, or sorry, strong regeneration is mainly due in part by the fact that it's so simple, right? Its head is a really simple head and it's able to make it again. Your head is a really complicated head and you're probably not going to be able to make it again before you die of bleeding, right? So these things they just have to make a new head before it starves to death. So things usually take about 3 weeks to starve to death, and if it can regrow back a head before then it'll be a new worm, right? So because it's so simple, its regenerative powers are really strong, and in fact, it can do asexual reproduction. Alright?
I had a zoology class where we actually cut them up and we saw how fast it grew, and it was just a matter of weeks.
Oh, really? Did you guys do like interesting cuts?
Yeah, we tried to like diagonal ones. But
Yeah,
they're so
yeah.
small.
Oh, okay, okay. Yeah, they're pretty small. How small were yours?
How small are they?
Yeah.
Like that.
Yeah, okay, that's pretty tiny. Yeah, I've seen flatworms at the beach before. They're only about an inch long. I've seen some of the big ones are about an inch, right? Maybe 2 inches. They're not huge, right? But yeah, you can cut them in all sorts of different ways, right? This is just some simple cuts. You take this one worm, cut its head off, cut its tail off. The midsection grows back a head and tail. The tail grows back a head, and the head grows back a tail, right? Now you got 3 worms. But look, you can cut them in interesting ways too. Like look at this. Here's some like different cuts you can do. Like if you cut off its head and destroy it, and then you take its remaining tail and snip it in half, then it'll grow into this. Yeah. So like, yeah, and over here, if you like cut off its head and destroy it, and then you cut this part in half, then it'll grow back 2 heads. So yeah, I don't know, they can do some crazy stuff, man. And then this is just the same as the one we just saw. Okay, so yeah, they have really strong regenerative powers, right? Growing back heads and stuff. Yeah, Tiffany?
So do you like purposely like in my cap in order to reproduce?
Oh, I see. I think for these worms, they don't. But if they ended up breaking in half, then they could reproduce, right? There are some organisms that purposefully break up and do that, right? So like strawberries, for example, right? Strawberries, they send a runner, a really long vine to another patch of soil, and then it gets cut off, and then now that one grows into a new one, right? So there are some that purposefully do that, right? I think sea anemones do that too, right? You got a sea anemone here, and they grow a tiny little sea anemone here, and it shoots it off, right? And then that one just starts growing, right? It starts growing tiny little sea anemones around on its body. So like if I grew like a small human right here, and then I just picked it off and threw it away, and then that one grew into it, kind of like that. It's called budding, right? Budding. So let's see, sea anemones, strawberries, and I think yeast does that too. Yeast is a type of fungus, right? So yeah, some of them do that. I don't think the worm purposefully does it, but if it did do that, then it would be a viable way of reproducing, right? Okay, yeah, so some can do it, but they don't do it willfully, right? Okay, cool, so Platyhelminthes, that's the first worm, right? Let's look at the second worm. Nemertea, all right, Nemertea. Phylum Nemertea is called a ribbon worm in the common name, and they are curly and smooth. That's as much as I want to talk about Nemertea. I'm not that excited about worms, to be honest. But it's good to know their names and to be able to tell them apart, right? Because, I mean, that's the best way to show someone that you know this stuff, right? Just start naming things. So, a curly and smooth worm is a Nemertea, right? That's all. Phylum Nemertea, ribbon worm. Alright, the next worm is a Sipuncula, Sipunculin, right? Phylum Sipuncula, the peanut worm. Right? You can find these in tide pools under rocks and stuff, you know, like in Monterey Bay, I flip a rock and I might find a couple of these Sipunculins underneath, right? So, you know, they don't really look like peanuts in my opinion, but they have a lopsided body, and depending on whether they move the fluid to one side or another, they can change shape and get longer and stuff. Right? So the peanut worm with a lopsided body is called Sipuncula, right? the next worm? The next worm, I'm gonna show you a clip first. All right, let's take a look at this clip first. I'm sure you guys have seen this before. What did he just call them? A nematode. Is that what an actual nematode looks like? No, this is what an actual nematode looks like, right? Nematodes are tiny, right? They're really small, and the reason why they always look clear like this is these are all microscope images, right? Because they're so small. You could tell them apart from other worms because they have this tapered end. Taper means it comes to a point, right? So you look under a microscope at a bunch of different worms, you find these clear ones that have a tapered end. Those are actual nematodes, right? A lot of them are parasites, but I don't really think they eat things like locusts, right? Not like that. And nematodes, a lot of parasites. They also don't age. And it's kind of strange. They don't get older, so they're under some intense research to learn their genes and how not to grow older. Okay, so this is an actual nematode, phylum Nematoda, right? A round worm with a tapered end. What those artists in SpongeBob drew is this. Phylum Annelida is known as the segmented worms. You guys see the resemblance now? It had lines on them. It was clearly an annelid, right? It was not a nematode, right? Segmented worms. Right, these guys have little segments to them. Okay, this is probably the most interesting group of worms in my opinion. They're a good bait too. But I have these 6 worms here with these letters. All right, I want you guys to take maybe 30 seconds, minute, split these worms into 2 groups. All right, and afterwards, let me know what you guys come up with. I'll give you guys some time. What do you guys think? Group 1? A, B, and E. Okay, and then that means that C, D, and F is in the other group? All right, what was your rationale?
A, B, and E have, it looks like pokey things.
They have pokey things. Okay, and the other ones don't. Okay, good, right? We're using the morphological species concept yet again, using our eyes to tell the difference because it's chemine, right? Okay, good. I would put them into those 2 groups as well, and so would scientists, right? Those ones have those hairs on the sides, and these ones seem to lack them, right? So those things are called bristles, actually, right? Bristles, and they're part of the class. Okay, so here we go. Phylum Annelida has several classes, and class Polychaeta, right? Polychaete worms, polychaetes, right? Polychaete worms, they have these bristles, right? The common name is called bristleworm. This is some stuff that I studied in my master's degree, right? Polychaete worms. But anyways, they got these bristles, right? Poly means many, and chaete means bristles, okay? So they got the many bristles, right? These bristle worms crawling around, right? Some people call them pile worms or lugworms or ragworms or a bunch of different types of worms, but bristle worms, right? And then sometimes they have scales on top, so you can call it a scale worm too. Okay, so class Polychaeta. You see these guys? These are kind of like feelers, kind of like legs. They probably walk around, right? Probably walk around on the ground. Well, not all polychaetes walk around. We have polychaetes that sit inside of a tube for their whole life, right? See these? These are tube worms. Tube worms are polychaetes that sit inside of a tube their whole life. See these? The deep sea tube worm? This white thing is a tube. Right? What do you think that tube is made out of? It's made of calcium carbonate, yeah. Okay, so there we go. Another calcareous organism, tube worms, a type of polychaete. A polychaete is a type of annelid, is a calcareous organism, right? So anyways, tube worms, right? We got 3 different types of tube worms here, like those deep sea tube worms, feather duster worms, right? This long part actually stays inside the tube, right? We just pulled it out. And Christmas tree worms, right? The ones in Avatar is based on Christmas tree worm. Okay? So let's look at this real quick. When you take it out, when you look at this thing, it doesn't even look like a worm. I mean, when you first look at this, I mean, like, what is this little Christmas tree or something? Like this little plant or whatever? It's blue, yellow. Right? It doesn't look like a worm, but when you take it out of its tube, it becomes more apparent that it's actually a worm. Do you guys see the bristles? Right? And the segments. So from the segments, you can tell it's an annelid. From the bristles, you can tell it's a polychaete. Right? So these tube worms, right? It turns out they're actually worms. And what do you think they use this thing for? They stick it out of their tube. Yep, it's a feeding structure, right? They use it to catch their food. Okay, so let's take a look, right? Let's compare the first polychaetes that we saw with the tube worm polychaetes, right? What is the difference between these two worms in terms of their lifestyle? Okay, they move, right? And these ones, they just stay in one place their whole life, right? So there are some animals that move around, okay? We call that being motile, right? Motile organisms move around, right? You guys are motile. You guys move around, or hopefully you guys are motile. Okay, well, if you guys made it to this classroom, I'd say you guys are motile enough, right? And other animals stay in one place their whole life, and we call those guys sessile. Okay? Can you guys think of another sessile animal or an organism? Doesn't have to be an animal. Sea anemone, how about a tree, right? Yeah, those things, those are sessile, right? Sessile organisms. Okay, so we got motile organisms that move around, the sessile organisms that choose to stay in one place their whole life. Right? We will talk a lot about sessile organisms when we go over the intertidal and how important it is for them to choose a good spot because they don't get any second chances usually. Right? Okay, but anyways, because some of them move around and because some of them do not move around, their feeding style is drastically going to be different, right? They're going to have to use a different strategy for eating. So let's look at the motile one, right? The one that is active and can move about. What do you think it's able to do if it's active and move about? Do what? Hunt. Yeah, it can hunt, right? Predation, right? It can go around and hunt live things, or it can eat dead things like scavenging, right? So you can hunt, predation. But what if you can't move? You can't hunt unless it literally comes right up to you, right? So what are you going to do if you can't move? What was that? Filter feed, exactly. If you're not going to move, the water around you will still move, right? And the water carries a lot of particles inside, plankton, okay? Catch those things and do your filter feeding, right? Filter feeding is the best strategy for eating in the ocean if you cannot move. Okay, so let's define these words, right? Define predation. Hunting for prey, right? That's what predation is. You expend energy, you chase them down, right? You go after them, hunt. Predation. On the contrary, very passive feeding, like traps. Filter feeding, right, is very passive, and they just sit in one place. They hold out a big net, and they wait for the water to pass through the net, and they catch whatever's in the water, right? Plankton, debris, right? Particles, dead things, right? Stuff like that. And hoping the water brings it in for them. Okay, so filter feeding uses a big old net and passes the water through it, gets the particles inside the water. And remember, that is the best way to get your food when you do not move, right? Filter feeding. As we'll see, almost every sessile organism is a filter feeder. You don't have to be sessile to filter feed. Right? There's some motile filter feeders out there. But if you want to be a predator, you should probably be motile. Okay. There are a few things that lie in wait for their prey, but most of the predators actually go around chasing them down. Okay, we all good with this? Yeah. Feeding styles as being dependent on their movement lifestyle, right? Okay, so I just want to introduce just one more term because these predators, right, the polychaete worms, if you zoom in on their head, they actually have a proboscis that extends out with claws at the end. So if you get a big enough polychaete, like one that's maybe like a couple inches long, 4 or 5 inches, it could actually bite you, which is, it's not gonna hurt, but it's just really creepy because I mean, like, I got bitten by a worm. That's creepy. Doesn't that sound creepy? I don't know, when you get bitten by something else, it sounds kind of normal, even though it probably causes more damage. This one doesn't hurt, but it's creepy, right? So notice how like it has these two fangs, right? These mandibles, okay? Whenever you get like a two-sided thing coming down, it's, you know, it's we kind of have a special word for that, coming down and grabbing something, like catching a prey by grabbing it, right? It's called raptorial feeding, right? Like a raptor. You guys know what a raptor is? It's a bird, right? Raptor is a bird, so they got those talons. Grab, okay? So when you grab your prey, that's how you catch it. Raptorial, right? Okay, and that's just one method of predation. Okay, is everybody doing okay so far? We're all good with annelids. I think we're almost finished here. Okay, so let's go into the other group, right? Remember, we probably already forgot about these ones, right? Well, we split those annelids into two groups. The ones with the pokey things, right? The bristles, the chaetae, right? And then the ones that did not, right? Oligochaeta, right? Subclass Oligochaeta. These are oligochaete worms. Do we recognize oligo now, right? What was the other oligo word that we know? Oligotrophic, right? Lacking nutrients, but this one lacking what are they lacking? The bristles, right? Lacking bristles, and they still have the segments, but they're a lot smoother, right? Okay, and this one is our terrestrial representative that we see every time it rains. Right? They all come to the surface, or else they'll drown, right? But yeah, we got these earthworms, and they got segments for sure, but they don't have the bristles because they are oligochaetes.
Why are why are they considered a class and this is a subclass?
Because oligochaetes are considered part of polychaetes actually, because they're closely related. Yeah, but that's a good question. Right, you know, it would make sense to put them in different, right? But they're pretty related. So. Yeah, so I guess not having the bristles is misleading. Okay, that's not completely correct. They're just reduced, and they're so small that, you know, they're when you look at it under a microscope, you can see them. They look like little hooks, so they still have them, right? They still have the bristles, but they're just really small. Okay, so all these guys, they got those tiny little hook-like bristles. They're not big and pronounced. Okay, cool. And then this one's not too important. I just want to introduce it. We will talk more about these later, but this is another type of worm called arrowworm, right? It's called Ketonatha, right? Ketonatha. Well, we know what ket means, right? Ket means bristle, and nath with a G, means jaws, right? So bristle jaws. If you guys look at the front, opens up its jaws and catches prey. What do you think its feeding style is? Raptorial, right? Because it grabs them like that. Okay, it catches. This thing's tiny, by the way, so it's like a microscopic plankton. Okay, so nothing huge. Yeah, we'll talk more about this when we actually talk about plankton, but I just wanted to throw that in there because it is a type of worm, right? Arrowworm. I mean, keep in mind it's not really related to the other groups. It's only a worm because it looks like a worm, right? It's just a, that's it, right? A long organism. Okay, everybody good with that? Okay, so it's a plankton, this is our tortoise. Alright, cool. So, here are all the different worms that we just talked about, right? 5 of them except for the Ketognath. Let's go all through all these things, alright? Name the phylum and tell me how you can tell them apart, alright? So, we'll start over here. What is this?
Peanut worm.
It's the peanut worm, but what is this? Phylum. Sipuncula. How do you know that's a sipuncula and those ones are not? It's got a lopsided body, right? Alright, let's go with this one. What's this? What was that? A ribbon worm, also known as Nemertea, right? A Nemertian. How do you know that's a Nemertian? It's curly and it's smooth, right? Pretty easy. What's this? Flatworm. Platyhelminthes. Yes. It's a flatworm because it's flat, but what else does it have? Yeah, it's got the eye spots right over there. Eye spots. What are eye spots good for? Yeah, detecting light and dark. They're just light sensitive cells, right? Okay, and what is this? That's a nematode. And it's a nematode because? Last but not least, this one. Oh yeah, I mean, yeah, it's called an earthworm, but what phylum are they in? Annelida, right? What does annelida mean? Segmented, right? So both of these things have segments. Can you guys further differentiate these two? This one is a bristleworm, a polychaete, and this one does not have the bristles, or doesn't have pronounced bristles, so it's called a oligochaete. Right? C-H-A-E-T-E, chaete. Okay, are we finished? Yeah, I think we're done. All right, so I want to go through the phylum that we just went over and something that I would like you guys to pay attention to whenever we go over phylum, okay? Okay, what would I like you guys to pay attention to whenever we go over a phylum? Okay, so notice when we went over the 3 plant phyla, we didn't really go into detail besides the fact that it was green and it has chlorophyll in it, right? That's all we talked about. We talked about its color and what gave it its color, and several examples. That's all. But for animal phylum, we're gonna get a lot more in detail. Still not like super crazy, this is still intro class, but we're still gonna get decently in detail with most of the different phyla out there, okay? So when we learn a phylum, okay, then to pay attention to, we should pay attention to the distinguishing characteristics or features, right? Features. And then that usually have something to do with the meaning of the phylum, right? Okay, so for example, Porifera, what was the meaning of that phylum? Has pores, right? Does that make sense? Okay, so the organism that has pores, and that happens to be the characteristics and features that distinguish it from the other phyla. Okay? All right, so distinguishing characteristic, this one. We should know its feeding style. Okay? You know, we should put Porifera down here on the side as an example of all these things, right? So phylum Porifera. Okay, distinguishing features, meaning, has pores, sponge. Feeding style, what is its feeding style? Filter. Filter feed. Okay? Next, we should know its larval type. what's the larval type of sponge? You know what it starts with? Starts with a P. Okay. Filter feeding is a feeding style. Parenchyma is a larval type. And we got some distinguishing features. Okay. We should also know its classes. Okay, and then attributes of each class, right? So classes of Porifera, we have like Hexactinellida, Calcarea. You guys tell me the last one. What was the last Porifera class? Demospongia. Anything else that we need to go over? I'd say these are the main things. There might be a couple other things that come about, but definitely focus on these things when we go over all the different phyla, right? The meaning, distinguishing characteristics, parts about its lifestyle, right? And then classes, all the different classes, right? I'd say the hard part, this is the hard part, right? A lot of crazy names, might get confused with phylum. I totally understand. You guys spend some time on it, you guys will get it. If they have a shell, you should know what a shell is made out of too. Right? So for example, not all sponges have a shell, but these two do. Right? And this one's made out of what? Silicon dioxide, and this one's made out of calcium carbonate. So if they have a shell, make sure you guys pay attention to that too. But other than that, I'd say these are the main things we should focus on when we go for phyla. All right, how are we doing? We all good? Anybody have any questions on this?